Hi everyone, welcome to Jack's Career Chats, a series where we highlight the diversity of jobs, skills, and opportunities in the world of biomedical research. Today, I have the pleasure of speaking with Nirja Katiyar, a bioinformatics analyst in the lab of Duigu Uchar at the Jackson Laboratories campus in Farmington, Connecticut. Nirja uses mouse models and the latest genomic technologies to study aging and cancer. Nirja, thank you so much for taking the time to chat with me today.
Thank you, Christina. It's my pleasure.
So why don't you start off with telling us a little bit about your journey to how you got here. So tell us the early days of you in school and studying and what drew you to the world of bioinformatics and genomics.
Yeah, sure. Thanks. So I grew up in India, and I did my undergraduate in biotechnology. So it's known as Bachelor's of Technology. It's a degree of 4 years. We had different specializations, and my specialization was in genetic engineering. During my undergrad, we had different courses. One of them was bioinformatics. And as I was always interested in programming, since my high school days, I was mostly interested in mathematics and programming, computer science. So I decided maybe it's a good opportunity to apply for a master's program and pursue master's in bioinformatics. So this is what brought me to United States.
So what did you like about programming at such an early age? I never had any programming experience in high school. So what was your interaction with programming at the high school level, and what drew you in undergrad?
Actually, I love programming during my school days. And I'm actually grateful that programming was introduced to us in our fifth grade itself.
Oh, wow.
We started with a language known as GW Basic. I don't know if anybody would have heard about that language in today's date. And it was a very basic language. It was easy to understand. And I just found it interesting. And then in our eighth grade, eighth and ninth grade, we learned C++. And then later we had Java in our high school. So because I always found programming to be interesting since fifth grade, I would say. I used to go to my teachers. I would ask them for more challenging problems apart from what would be given to us in a normal homework, because I wanted to explore more and I was really excited. And I do remember that I realized that how passionate I was at that time for programming. I would not go off to sleep till I was able to solve the program. So I have to write the program and get it working till even if it's 1 a.m. or late night. And I'm glad at that time my dad was able to get, it was a used computer, black and white, from somewhere for me. So I was very happy and very excited that, okay, I have this at home and I can program as long as I want.
So what were your programming projects like at such a young age? Is it like a puzzle? You said you had to, you were working to figure something out. What did it look like? What were you doing?
Yeah, so those at that time, they were more basic programs. You would have to generate a series, everybody would be knowing now, Fibonacci series. And then you would have to generate a pattern, like a tree of stars. So different kind of patterns using different characters on our keyboard. So yeah, all different kind of programs. Now it's difficult to remember.
Probably been a while.
Yeah, it has been a long time, but it was fun.
Yeah. So that was really influential to you to pursuing bioinformatics and biotechnology classes in college, you said, in undergrad.
And even before coming to US, I had some free time. So after I had applied and I was waiting to hear back from universities here. So at that time, I also went to a different city in India to pursue another certification of Java. So I could learn more and just polish my skills. So there we had more proper projects where I would be building a website and with a database in the background with usernames and passwords, so you can log in, and only people who have a account in that particular profile would be able to log in, and otherwise it will be rejected. So that was a fun project, I remember.
Yeah. What motivated you to want to pursue a graduate degree? So you pursued, you said, your master's in the US. So after undergrad, did you hold any jobs after undergrad, or did you move straight into grad school?
So I moved straight to grad school. During my undergrad, I had other friends who were preparing for MBA, and some of them were preparing for GRE and TOEFL. So I was living in a hostel, and my roommates were also preparing for GRE and TOEFL. So they were good friends, and some of them were even in my high school. So it was a good bonding, and it does influence you, like your friends around you. They do influence your decision. I was considering also like keeping an option for MBA in India. But finally, I would say my love for programming brought me to US for the master's in bioinformatics program. And in India, we really did not have many programs in bioinformatics at that time. So it was important to look for an opportunity where there are good programs to offer.
I see. So because you chose the field bioinformatics for your graduate studies, it was important for you to find obviously programs within that. And so the best opportunities were in the US.
Yeah.
That must have been really difficult to leave your family to study. How was that for you, deciding to leave home and to come very far to the US for your studies?
Yeah, that's a good question. I had honestly never dreamt during my childhood days that I would ever leave India. That was never on my on my plans. But I think it was my dad and my uncle who actually were biggest supporters and who influenced me that I should apply and apply to United States. I actually also applied for some universities in UK, and I did get admit. But at the end, when I had to make my decision, I made my decision with the help of my family, and I think coming to United States was a good choice.
Yeah, how wonderful to have the support and encouragement of your family. How was your experience in grad school different from undergrad? So now you really immersed in the field of bioinformatics, and your master's program gives you a more in-depth coursework. Do you think that was an important step to prepare you for your next jobs? Were the courses different? How was grad school different than undergrad?
Yeah, grad school was quite different, I would say. It could be some difference in the education system in United States compared to India as well. But I really enjoyed the experience, I have to say. I made good friends in my graduate school, and the professors were also very helpful. We had different programming classes as well as other classes related to proteomics and genomics. Um, and one big thing that's different was we have open book exams here in US, which I had never heard of before when I was in India. So that was a new concept to me. And yeah, it was fun to work on assignments and prepare for midterm exams. And also, like for assignments, we would be able to have some discussions with friends. There were some group projects, which were also very, very, very helpful. It's good to have to work in a team and learn a lot from each other. So I think it was a fun experience.
And what kind of work were you able to do during your schooling, both in undergrad and in grad school? Did you work in a laboratory? Did you work on research projects, or was it just all school work?
So during my undergrad, I also did some internships. And for internships, there are some research institutes in New Delhi in India, and also in my hometown, which is Lucknow. And I went through a couple of internships, learning the wet lab work to do experiments like pipetting, isolation of DNA from bacteria or isolation of RNA. I remember from like plants, mango leaves. So those were like small mini projects which I was doing during my internship and learning in the process. And my undergrad dissertation was in bioinformatics. I decided to go to a different city known as Chandigarh. It's a beautiful city in India, and yeah, I was excited. So it was a good experience. And during my for my undergrad dissertation, I worked on designing. It's a drug designing project for Clostridium botulinum. It is a bacteria for food poisoning. So it was a very interesting project, and I learned about docking and how what all processes are involved, modeling for drug designing process.
And how would you use computational techniques to do your research?
So, so regarding my undergraduate dissertation, it was in two parts. I just spoke about one part, which was the drug designing project. The other was a database, and it was a MySQL database, which was on restriction enzymes. So we use computational techniques like I was using Perl and MySQL uh, to design the database, as well as develop the front end, which was a website where any user could enter a restriction enzyme information and try to extract a lot of information from the database. The backend was in MySQL and the front end was in Perl with HTML. So these were the programming languages that we would use. And for drug designing, it's more about just learning how to run different applications and programs that have already been developed. But I can tell you more about masters, I guess.
Yeah, no, it sounds really interesting, and it sounds like you had a really a good variety of experiences, not only just in one aspect of biotechnology and computational space. I mean, you did pretty much anything from, you know, designing a website to modeling drugs and everything in between. That sounds really fascinating and probably a wonderful experience to set you up on the research track that you're on now.
Yeah, no, that was a very, very helpful experience. And my teachers of bioinformatics were very, very helpful. So I have personally noticed that if I find good teachers of any subject, it really helps me and motivates me to develop interest in that particular subject. And I think it also has a role to play in influencing my decision about my career and my interest and my passion.
Yeah, absolutely.
Yeah. And during my master's, I also did internship. And I was selected at the J. Craig Venter Institute in Maryland. And that was a really good opportunity. So I'm grateful I got that.
Yeah,
To
that's
be
a
honest,
leading genomics institute, so lots of good genetics research happening there as well.
yeah, yeah, it was a very good opportunity. So I'm grateful that I got that opportunity. And I also worked on a variety of projects with three different mentors throughout the year, like different parts of the year on different projects. And I got experience working on like protein-protein interaction data. And then even with the next generation sequencing data, that was the first time that I started working with our next generation sequencing data, writing programs for basic stuff that my supervisor wanted me to for internship. And then I also worked on a metagenomics, using a metagenomics software on a metagenomics project. So that also led to me to be a co-author in one of the publications. So that was very, very helpful. And I'm still in contact with my mentors who were during my internship. So they have been quite supportive.
Oh, that's special. So you've cultivated these career mentors along the way that have been able to support your career.
Yeah, now one of them is at NIH. So it helps.
Yeah, awesome. And so after grad school, what were your next steps after your master's? Did you apply for jobs, or were you thinking about doing a PhD? What were your steps during master's and afterwards?
Yeah, so I was thinking about both, but I realized that gaining some work experience would help uh, to make the decision better. It will be easier to make the decision. And then I went to I worked as a like a research assistant. I was in Penn State for 3 years, and I worked with different professors in different labs and gained a lot of experience and also publications in the process. And after that, I moved on to California, and which was good. And I got the opportunity to work in the core facility, core bioinformatics facility, and I was the only bioinformatician at the core facility. And that gave me a lot of experience, like practical experience, how busy life can get as a bioinformatician.
Yeah, so in a core facility, that's somewhere that the labs around will use the services of the facility and the staff like yourself, like a bioinformatics analyst, to help them analyze their data, make sense of it in order for them to pursue their research.
Yes. Yeah, so we were providing service. We were also coordinating with our the genomics core facility. So all the data that would be sequenced in the genomics core would would be collected by us, and I would do the quality control and demultiplexing, and then update our database and send an email out to the professors and different labs that their data is available to download and make it available on the website.
So it sounds like a ton of skills, not only the hard skills of, just knowing the coding and the programming and the genomics and genetics, but also a lot of skills like time management, because you had all these projects, communication with all these different types of people. So it sounds like you got a ton of experience.
Yeah, I have to say, I think I got the most experience there. Apart from, so this is something which I was just doing on the side, providing the data to everybody after quality control and demultiplexing. My major projects that I had to work on were actually real bioinformatics projects. So I was supposed to work on RNA-seq projects, DNA methylation projects, ChIP-seq. So we would get a variety of projects from different PIs, and they would ask us for our service. So that was one of my major job responsibility. And the other responsibility was to also teach workshops. So I had to prepare for teaching workshops on different topics, and also support our core facility and our director of the core facility. And I would also generate the certificates and send them out to the participants and work on the registration form and all kinds of miscellaneous activities.
Wow. So in addition to all of the bioinformatics work, you've also had some teaching responsibilities as well.
Yes,
Were
yes.
you trained in teaching? Had you had teaching experience before, or was this your first dive into it?
Uh, so no, I did not have any professional teaching training, but yeah, all I can say is like since my childhood days, I would say I used to teach my younger brother and sister at home, and my friends in high school, because they knew that I love programming, so they would all come to me for programming help, and even my brother and sister along with my friends. And even during my undergrad, although my degree was in biotechnology, I was still helping out some friends who were pursuing degree in computational sciences.
Mhm.
I think I loved programming, and I think I loved teaching as well. So I really enjoyed that experience. Even when I had to make the decision if I should be moving to California or I should join another job at New York, where I had an offer. The reason why I moved to California was because the director of the core facility there was able to um, he was able to convince me that because of my love for teaching, he was like, you will get all these opportunities here. And I was like, yes, that's true. Okay, I'm coming to California.
Yeah, that's great that it was so influential. It means that you're so passionate about it. And what I really love hearing is that you didn't necessarily have formal training, but that it's okay because of your passion and because of your skill, because you would incorporate it, into your life anyway. That was, kind of enough experience to get you to that job.
Yeah, definitely. And it helps if you prepare presentations and teaching material. And also, I also attended. So the first year when I had joined in the month of October in California, and I was supposed to teach the workshop in December. And the first 4 days were taught by my director. So I was able to see how he was teaching. And I think it does help you when you focus on how your professors are teaching you and you're able to generate interest in the subject. So
Yeah, great. So now you work at Jax as a bioinformatics analyst. So it sounds like a similar job title to what you've had before, but now you're working in aging and cancer. So probably the field has changed a little bit in terms of the subject matter of what you know. So can you describe a little bit about the type of work that you do now?
yeah, sure. So before coming to Jax, like at in California, especially at UCR, all the work was related to plants. Everybody was working on plants mostly there, but I was also more interest in human research, human disease research. So I'm actually very glad to be at Jax. And here, my PI, Dr. Dugyo Uchar, she works with aging. And currently I'm working on a project in collaboration with Dr. Olga. And her research experience, her focus is on cancer. So we are looking at breast cancer, and we're looking at relationship between aging and cancer. And for this, we're using mouse models for our research.
And so you're sequencing the different types of mice that they have, these cancer models and the aging models, and you're using all of that genomics data to do the research that you're doing.
Yes, yes. Yeah, and it's very exciting. I'm working using the single cell technology, single cell RNA-seq and single cell ATAC-seq technology, which is kind of the hot technology in our field right now.
So that's taking, usually we dissect like a tissue from an animal and you will process the DNA or RNA from all the cells in that piece of tissue. But now this is taking that tissue and breaking it up into single cells and looking at from this one cell, what's the DNA or RNA profile, from the next cell, what's that profile.
Yeah.
That sounds much, I know it's much more computationally intensive than a bulk RNA-seq or bulk genomics.
Yeah, so there are many machine learning algorithms that we use in the background to be able to analyze our data. And we have to perform clustering. We have to look at different patterns in different cell types because we are doing this analysis at cell level. So it is quite interesting and exciting. And also after coming to Jax, I was able to attend the cancer course, which was head by Dr. Karina Paluka. And it was an amazing course. So I think it was a great opportunity for me and the perfect timing to learn about cancer. And now I'm able to relate aging and cancer for
It
my.
sounds like, yeah, and it sounds like in every job, it hasn't just been a job. You've been able to learn so many skills, but also a little bit into new fields. Like now you said, you're diving into cancer and immunology. How has that experience been for you? It seems like you like learning, so I feel like it's a good experience, but can you just describe the opportunities that you've had to just kind of do that professional growth and development and learn more along the way?
Yeah, I honestly love learning and teaching. So I think I will always look forward to whenever I'm moving from one job to the other. I think the most important thing that I will be looking forward to is be able to gain more skills, new skills. That is what keeps me going. So, and you stay excited. Okay, you have something new every day. And so it has been a good experience. And like my master's in bioinformatics was mostly focused on proteomics. So we did not really have much of genomics. And when I did my internship, that was the first time I kind of touched genomics. And then at Penn State, all my work was on genomics related projects. So it was like genome sequencing. It was one project was on copy number variation for autism kids. Yeah, so there were a variety of projects that I was working on, and all these different experiences really helped me keep going and like staying excited about my field.
Has that ever been a barrier for you that, you know, if you're jumping to a different subfield like autism versus cancer or something that you don't really know the background and so you have to learn it?
So, I mean, of there is no doubt learning anything new is a little bit of challenge for sure, be it different fields in biology or different programming languages. So there are challenges, I would say. Like when I was in Penn State, I was coding in Python mostly. But when I moved to UC Riverside, our director wanted me to completely switch to R programming. And that was a bit of a challenge, to be honest, because I feel all programming languages except R, from my experience, you have your thinking mode is in one way, and R you have to completely think in a different way. So that was one. I was very surprised that I found it challenging, but it was a bit of challenge in the beginning because I had learned so many programming languages before. I never thought I would find it challenging, but there are times when you get surprises. And yeah, regarding autism, I think that was still okay. Yes, copy number variation, learning about it was a bit, it's different, but I'm finding it very exciting to learn more immunology here. And I found that this was, initially I really felt like, oh, I wish I knew more. But I'm so grateful to my supervisor. She's so supportive. Like she's always encouraging, and she's always completely fine if you don't know this thing. She's like, you will learn with time. And I just love that, the quality in her, that she's always supportive and keeps encouraging about learning new things.
Yeah, that's really wonderful and so essential to success, right? To have that encouragement and the space to learn and to grow.
Yeah, and I strongly feel that if you get good mentors and good teachers, be it in high school, undergrad, graduate, or later during your job, if you have a good manager and a good mentor, it can really take you to places. So I feel mentors have a big role to play. And that's why I also feel that I also want to be a good mentor for others, so I can help out as many people as I can.
Yeah, absolutely. I mean, you've shared such good pieces of advice now. And also, what I liked about what you said is just sharing that you didn't know everything for every step of your projects or for every job that you have had, and that was okay, and that you had the time to learn, and you know, and it's expected. You don't have to know everything going into a job, and you know, even if you have a bachelor's or a master's, you might not know something when you come to your next job, and that's okay as long as you can learn and you can adapt.
Yes, I completely agree with that.
So who are the types of people that you work with in your jobs? So you're at a research facility and you're in a research working group. Are there a lot of other bioinformaticians like yourself that are on the dry lab? Do you work also with wet lab? What are your colleagues like?
Yeah, so my lab is mostly computational. So my other co-workers, they are all dry lab people. Um, and we have graduate students whom, so one of my responsibility here is also to help them out. So I do help.
You're teaching there in mentorship.
Yeah. Although, I mean, I'm not teaching proper official workshops right now, but I'm still helping out other graduate students who are joining our lab, who may have questions of any kind. And we also have other bioinformaticians who are working on different research projects, but we all learn from each other, and we all provide feedback to each other.
That sounds like a very collaborative environment, very supportive. There's a lot of communication and sharing going on.
Yeah, yeah. And of course, so the other project I was working on at JAX was on promoter capture high C, which is also something I had never worked on before. So that was also very exciting. So it's good when you get to know that you're working on something which very few people might have worked on. So that's good. So for Promoter Capture High CS, we were collaborating also with wet lab people, because the people who are doing the experiments, we would meet them on a bi-weekly basis and have discussions, and I would present the analysis to them. And that's how, and it's the same with our aging and cancer project as well. And our collaborator and her postdoc, they are working on the experiments on the mouse models, and they are at Bar Harbor facility. Like the experiments are done in the Bar Harbor facility. And then we have bi-weekly meetings where I present the analysis of the projects. So it's very helpful actually, because I learn a lot of biology from them, and they are able to learn some programming from me. So it's a good collaborative effort.
Yeah, it sounds like a very good back and forth working group. You both gain from each other, both sides.
Yeah.
And so what does the work look like that you're doing? Before you talked about how some of it was, algorithms, some of it was website design. What are you doing now with your genomics sequencing projects?
So I'm basically analyzing the data. So analysis of the data starts from performing basic quality control of the data.
So the data is the sequencing, right? So the reads of ACTG sequences, millions and millions of them, right?
Right. Yes. So we receive the sequencing data from the sequencing core facility at JAX, and then we perform the quality control. We look at different parameters, if we have sufficient number of reads and the read depth for each individual samples. And then there are a long list of parameters that we have to check, especially for single cell. So there's a tool known as Sura. It's an R package. It was developed by Dr. Rahul Satija's lab at New York Genome Center. So we are basically using that package in R to analyze the data. And there are multiple steps involving clustering, generating heat maps for visualization of the data, looking at finding differentially expressed genes between cell types, and also within the same cell type at different ages. So we look at different parameters that we are interested in for our project, and generate models accordingly, and look at the results.
Yeah, great. So you're using a lot of genomic software to help you analyze the data. These are programs that were already developed. And it sounds like you're also working a lot in visualization to figure out how to convey the information in your research findings, what graphs are appropriate, how to display your information.
Yeah. So yeah, one thing I would definitely mention, that if anybody is interested in pursuing a degree in bioinformatics or a job in bioinformatics, they should definitely focus on programming skills and statistics. And visualization is a big thing right now. So people can develop apps, they call it shiny apps, and develop websites, and also different kind of graphs. So you can generate good, variety of graphs in using programming languages in R or Python, using different libraries. So that is something, a very good skill to have nowadays, and it's very important to have for any research project.
Yeah, thank you for sharing that. I think that's really important to just, for any aspiring, bioinformatician or someone in data science or even in genetics that is kind of interested in programming, what words of advice and things that they should look out for. So that was great. Thanks for sharing that.
Thank you.
Do you know what's next for you? What are you thinking of? In your career journey, are you happy where you are? Are you looking to pursue a higher degree or a different type of job in the future that you would really like to see yourself?
So I do love working at JAX. Currently, I have just completed 1 year. And I also like my mentor, my PI, Dr. Ucha. So I think I would like to work with her and learn more about aging and cancer and immunology. So I gain more experience in the field. So it's good to work in a field for some time, so you also gain an expertise before moving on to a different field. So it's good to
Great.
establish
Yeah.
yourself at one place, I would say. So you gain more confidence and you can show your skill set. Um, and yeah, in future, I've been thinking on the side of if there is an option for a, I don't know if there is an option for a part-time PhD or something, but I'm just trying to explore the options right now. But otherwise, I think I would like to continue working here for some time so I can gain more expertise in this field because I really enjoy it.
Yeah, great. And I really like what you said about your advice for trying to stay in one field for a good amount of time, just to help yourself develop that expertise and probably give you a leg up for the next position that you have, that you're not bouncing around too much too soon.
Yeah, it's good to take some time so you understand where that also helps you to decide what should be the next step.
Yeah. Do you have any kind of parting words or final words of wisdom? You gave some great tidbits before, so you might not have anything, but if you have anything to kind of close off, I think this was a really great interview. You shared so many great points that are really valuable, valuable insight from someone with your experience. And I think that it's really an inspiration for a lot of students that would be pursuing computational computational work in the future.
Yeah, I mean, I think I would, yeah, I think I would just say that it's important to have good mentors, and if you have a good mentor, then try to stick to the mentor and learn as much as you can. And yeah, for bioinformatics, again, statistics and programming are two very important things to keep in mind and learn. And nowadays in research, as we all know, we can Google things up and we are all learning. Even if we don't find any proper courses, there are so many courses available on Coursera, LinkedIn, or Udemy, and there are so many courses available. I even go to YouTube many times to attend lectures from different professors in universities related to different topics, like any new topic that I would be working on. It's very helpful, I would say. And I think it's important to just keep learning new things and stay motivated and keep growing.
Yeah, it's good to hear too that learning doesn't have to come from school. I mean, school's very important, obviously, and education opens doors, but you can be in charge of your own learning. And there are so many free resources out there as well to always develop yourself and your interests and your skills.
Yeah. So yeah, I think that's all I have to say.
Yeah, no, that's great. That's great. Was there anything that we didn't get to talk about that you wanted to talk about? Because otherwise, I'll close it out. I think it was a
I'll
really
just
solid.
add one more thing. Somebody contacted me for help. So I gave one small piece of advice that whenever you have job interviews, if possible, you can try to note down the questions asked in job interviews and prepare a Google Doc so you can refer to it in future. And it really helps to prepare yourself for future job interviews.
Yeah, that's excellent advice. So you never forget. You're always reminded.
Yeah, that's all.
Wonderful. Well, thank you so much, Nirja, for taking the time to chat with us and share all of this information. It's really valuable. I've really learned a lot from your experiences.
Thank you, Christina. Thank you.
And thanks to everyone watching. Thanks for tuning into this episode. Don't forget, we have a whole bunch more of interviews on the Career Chat series, so be sure to check them out. Until next time, bye-bye.